The development and organization of peripheral lymphoid tissues is controlled by cytokines and chemokines. Surprisingly, members of the TNF and TNFR family, which had been thought to be involved in inflammation and cell death, are also critical for normal lymphoid development. This has been best demonstrated by a series of knockout mice in which either the ligand or receptor has been inactivated. As shown, these knockouts have complicated phenotypes. This is partly due to the fact that individual TNF family ligands can bind to multiple receptors, and conversely, many receptors can bind more than one ligand. In addition, it seems clear that there is some overlapping function or cooperation between ligands. Nonetheless, some general conclusions can be drawn. Lymph node development is dependent on the expression of lymphotoxin family members, LT family members. Interestingly, not all lymph nodes are dependent on the same signals from these family members. LT alpha 3, a soluble homotrimer of the LT alpha protein chain, supports the development of cervical and mesenteric lymph nodes and possibly lumbar and sacral lymph nodes. All of these lymph nodes drain mucosal sites. LT alpha 3 probably exerts these effects by binding to the TNFR1 and possibly also another TNFR family member called HVEM. The membrane-bound heterotrimer comprised of LT alpha and the distinct protein LT beta binds only to the LT beta receptor and supports the development of all the other lymph nodes. In addition, Peyer's patches do not form in the absence of the membrane-bound LT heterotrimer. These effects are not reversible in adult animals, and in fact, it can be shown that there are certain critical developmental periods during which the absence or inhibition of these LT family members will irrevocably prevent the development of lymph nodes and Peyer's patches. And that's the development and organization of peripheral lymphoid tissues is controlled by cytokines and chemokines. Have a great day.